Welcome back to the deep dive. You know, there's this really comforting idea that we tend to tell ourselves about disease. This idea that it's mostly just, well, bad luck.
Right, the whole bad roll of the dice theory.
Exactly. Like you play the short straw, you get a random mutation in your DNA, and then things just go wrong.
Yeah, but there is a massive, massive hole in that theory.
Right, and that's what we're looking at today. Why is it that the exact same literally the exact same genetic spark will completely burn the house down if it happens in the pancreas?
But if it happens in the lung or, say, the intestine, it barely causes a flicker. It might not do anything at all.
It's wild. And this is the central mystery we are unpacking today.
It really is. It's the classic seed and soil debate in oncology. For 50 years, we've been absolutely obsessed with the seed, the mutation itself.
Finding the bad gene.
Exactly. But today, we are going to talk about the soil, the environment that gene lands in.
And we are doing that by going through a massive new study from Nature, published just this year in 2026. These researchers, they didn't just write another paper. They actually built something called the Mouse Cancer Cell Line Atlas, or MCCA for short.
Yeah, it's basically a library, but not just a static list of books. It's a dynamic map of 590 different cancer histories, and it kind of completely rewrites the rules on why a tumor grows in one organ but dies in another.
So our mission for this deep dive is to decode those rules. We are going to go concept by concept through the logic of this paper. We'll see how a hidden ghost in the genetics has actually been ruining drug trials for years.
That part is fascinating.
It really is. And we'll meet the villain of the story, KRAS. And then finally, we're going to watch a sort of three-act play comparing the pancreas, the intestine, and the lung to see exactly who survives and why.
Let's dive right into it.
Okay, so let's start with the tool itself, the MCCA. Why did they even need to build this? Because frankly, I assumed we already had freezers full of mouse cancer cells in labs all over the world.
Well, we did, but they were a total mess. One lab in Boston might have 5 cell lines, and another lab in Tokyo has 10. And they've been growing separately in these little isolated dishes for decades.
Oh, so they're totally unstandardized.
Completely.
Yeah.
They undergo genetic drift. And because mice are the primary way we test drugs before we ever put them into humans, that fragmentation is really dangerous. You can't compare apples to apples.
Right, so this team centralized everything. 590 lines from 81 different mouse models.
Yeah.
And they didn't just collect them. They sequenced the DNA, the RNA, they photographed them.
They did everything. MCCA for the genetics, morphological profiles. They tracked clinical data like metastasis rates. But when they did that deep sequencing, they found something incredibly disturbing.
A bug in the system.
A huge bug. One that might explain why so many cancer drugs seem to work miraculously in mice but then fail completely in humans.
Okay, this is the immunophenotyping section of the paper, and you have to walk me through this because it reads like a detective story.
It really does. So to test a cancer drug, you usually take cancer cells and you inject them into a living mouse to see if the tumor grows or shrinks, right?
Right.
But you can't just put any cell into any mouse. The immune system is incredibly smart. If the genetics of the cell don't perfectly match the genetics of the mouse, the immune system rejects the tumor.
Just like an organ transplant rejection in humans.
Exactly. So standard procedure is to use a very specific purebred lab mouse called the C57BL6, or just Black 6 for short.
Okay, Black 6, so you assume your cancer cells are also pure Black 6.
That is the assumption everyone makes. But these researchers developed a really sensitive tool to scan SNPs. Those are single nucleotide polymorphisms.
Just tiny variations in the DNA,
Yeah.
like genetic fingerprints.
Exactly. And when they scanned them, they found that a huge number of these supposedly Black 6 cancer lines were actually imposters.
Imposters how? Like they were the wrong mouse entirely.
Not entirely. They contained huge chunks of DNA from a completely different mouse strain, the 129 strain, which is a brown mouse.
Wait, pause right there. How on earth does brown mouse DNA get into a black mouse cancer line? Did someone just mix up the cages in the lab?
No. It's actually a historical accident of how we engineer genetics. When scientists first create a cancer gene in the lab, say a mutant KRAS or P53 gene, they often do that engineering in stem cells from that 129 strain brown mouse.
Why the brown mouse?
It's just easier to work with structurally. The stem cells are more robust. But then They take those engineered brown mice and they breed them with black 6 mice for generations and generations.
Trying to wash out the brown DNA.
Right. They want a pure black 6 mouse that just happens to have that one single engineered cancer gene.
But they didn't get it all.
No. Because of physical proximity on the chromosome, the DNA sitting physically right next to the cancer gene hitchhikes, it survives the washing out process. We call them passenger SNPs.
Oh, wow. So you have this black 6 mouse that looks totally normal, but right around the tumor gene, on a microscopic level, it's wearing a brown mouse costume.
Exactly. And when you take those cancer cells and put them into a truly pure black 6 mouse for a drug trial, the mouse's immune system sees that costume.
And it attacks.
Vigorously. But here's the kicker. Imagine you are a scientist testing a new immunotherapy drug. You give the drug, the tumor shrinks rapidly, and you think, Eureka, my drug is amazing. The immune system is killing the cancer.
Oh, no. But actually, it was just the mouse rejecting the transplant because of those hidden brown mouse genes.
Exactly. The drug might have done absolutely nothing. The study calls this effective PTMB, protein-altering tumor mutation burden. It creates a completely false signal of efficacy.
That alone is a huge revelation. It means we might have approved drugs or even rejected drugs based on a complete misunderstanding of what was actually happening in the mouse.
It forces a lot of retrospection in the field, yeah. But the good news is this atlas finally maps all those ghosts. So now scientists know exactly which lines to use and which to avoid so they don't fall into that trap.
Okay, so they cleaned up the map. And once they did that, they could finally look at the actual biology. Which brings us to the villain of our story.
KRAS, the most famous bad guy in all of cancer biology.
It's the most frequently mutated oncogene, right?
By far.
Now, I've always been taught, and I think a lot of people listening have been taught, that mutations are binary. It's a switch. You have the mutated gene or you don't. The switch is on or it's off.
Right. That is the classic textbook view. But this study conclusively shows that it's wrong. It's not a switch at all. It's a volume knob. They focused heavily on a concept called allelic imbalance.
Okay, let's break down the terminology they use here, because they divide this into 3 specific categories that are super important for the rest of the paper.
Sure. So remember, you have 2 copies of every gene, one from mom, one from dad. The first category they looked for is called DGD, decreased gene dosage.
Meaning what?
Meaning the tumor cell actually has more wild-type, healthy KRAS than it does mutant KRAS.
And what did they find with DGD?
They found it basically never happens. Cancer absolutely hates that state. It provides 0 evolutionary advantage, so those cells just die out.
Okay, what's the second category?
HET, heterozygous. That's your perfectly balanced 1-to-1 ratio, 1 healthy copy, 1 mutant copy.
And then the big one, the third category.
Right, IGD, increased gene dosage.
This is where things get nasty.
Very. This is where the tumor gets incredibly aggressive and realizes 1 mutant copy isn't enough, so it amplifies it. It makes 2, 3, 4 copies of the mutant gene, or it just deletes the healthy copy entirely to remove any competition.
So it's basically cranking the mutant volume up to 11.
Exactly. And the clinical data backs this up perfectly. Patients whose tumors have reached IGD, this high mutant dosage state, have significantly worse survival rates. The tumor actively wants to reach this state to thrive. But here is where it gets really interesting and where the whole seed and soil thing comes into play. The tumor wants to turn up the volume, but the study shows it can't always just do it whenever it wants. The soil, the specific organ tissue it's living in, completely dictates when or even if that volume knob is allowed to get turned up.
Yes, and this leads us right into our 3-act play. We're going to look at the pancreas, the intestine, and the lung to see how different tissues react to KRAS.
Okay, before we start the play, let's just do a really quick 5-minute recap of the setup for you listening at home. Point 1, they built the MCCA, this massive library of 590 cell lines, to fix the fragmentation problem.
Point 2, they discovered passenger SNPs, ghost DNA from brown mice hiding inside black mice that causes fake immune responses and messes up drug trials.
And point 3, KRA isn't just on or off. Tumors actively strive for IGD increased gene dosage to become more aggressive and survive.
Perfect summary. So let's jump into Act 1, the pancreas. I always think of the pancreas as this really quiet, angry organ. Like, you don't mess with it. What is the environment actually like for a cell living there?
So the specific cells we are talking about here are the acinar cells. I want you to visualize them as workers in a highly, highly specialized factory.
Okay, factory workers.
Their entire job, day in and day out, is to just pump out digestive enzymes. They are what we call terminally differentiated, meaning they have picked a career and they are sticking to it. They do not want to divide, and they absolutely do not want to change their identity.
So if I'm a mutant KRAS gene, and my goal is to start a riot in this factory and build a tumor, what do I actually have to do?
You have to force the cell to have a complete identity crisis. It has to stop being a factory worker, quit its job, and go back to being a student. A generic stem cell that can divide rapidly.
Going back to school.
Yeah.
The technical term for that is de-differentiation. Right?
Exactly, de-differentiation. But the researchers found that just having a little bit of mutant KRAS, that HGT state we talked about, is not enough to convince the worker to quit.
So a low-volume KRAS doesn't work. How did they prove that?
It's brilliant. They ran an experiment where they used a chemical switch called doxycycline to literally drip feed different levels of KRAS into the cells. They created an artificial volume knob.
Wow.
And when the KRAS volume was low, the factory just kept running. The cell completely ignored the mutation. Markers like CPAR2, which is a classic factory worker gene, stayed super high.
But when they cranked the KRAS volume up to IgD levels?
The cell lost its mind. It lost its identity. That factory gene, CPAR2, completely plummeted, and stem cell markers like SOX9 skyrocketed. The worker went back to school.
So high volume equals identity change. Simple enough, just crank it up.
But there's a catch. A massive, massive barrier standing in the way.
The police.
The police. Specifically, a major tumor suppressor gene called CDKN2A.
Okay, so what does the police station look like in a healthy pancreas?
It is wide awake and fully staffed. To understand this, we have to look at the chromatin.
That's the physical packaging of the DNA, right?
Yeah.
How tightly it's spooled up.
Exactly. In the healthy pancreas, the chromatin around the CDKN2A gene is marked with a chemical flag called H3K4MN3.
H3K4ME3.
And that is the universal chemical flag for active, right?
Correct. It's like a giant neon sticky note that says, read me now. It means the gene is totally accessible and ready to fire at a moment's notice. So the very second you crank up the KRAS volume to try and force that identity change, CDKN2A senses the stress.
And the police raid the factory.
They arrest the cell immediately. They trigger a state called senescence. It's a permanent biological timeout. The cell doesn't die, but it can never, ever divide again. Game over for the cancer.
Oh, wow. So the tumor has a sequencing problem here. It needs high KRAS to change the cell's identity, but high KRAS instantly triggers the police to shut everything down.
Exactly. So the evolutionary choreography, the dance the tumor has to do, is incredibly rigid. Step 1. You have to kill the police. You absolutely must mutate or delete CDKN2A first.
Get rid of the barrier. Right. And then, and only then, step 2. You amplify KRAS to IgD levels to force the dedifferentiation. If you try to amplify KRAS before the police are dead, the cell shuts down and the tumor fails.
That is so precise. And does the real-world clinical data actually back up that exact sequence?
Completely. In human pancreatic cancer, 82% of tumors have lost CDKN2A. It is a hard biological requirement to get the disease going.
Okay, so if you're keeping track, pancreas equals strict police. You have to break the law, delete the suppressor before you can start the riot. That's Act 1. Now let's move to Act 2, the intestine.
The intestine is just a whole different ballgame. It is the polar opposite of the pancreas. It is not a static, quiet factory. It is a high-speed conveyor belt.
Because the gut lining is constantly shedding, right? We're always replacing those cells.
Constantly. The stem cells live at the very bottom of these little valleys called crypts. They divide, and then they physically move up the walls of the intestine, up these finger-like projections called villi, like an escalator.
And then what happens at the top?
Within a few days, they reach the tip, they fall off, and they die. They are shed right into the gut lumen and flushed out of the body.
That seems like a total nightmare for a cancer cell.
Right.
I mean, I get a mutation, I start to grow a little tumor, and whoops, I just fell off the escalator.
That is exactly the fundamental problem a tumor faces in the gut. The study showed that KRAS, even if you crank it all the way up to high-dosage IgD, cannot stop that conveyor belt. It cannot stop the cell from differentiating and being flushed away.
So the winning strategy from the pancreas, just crank up KRAS to force an identity change, is completely useless here.
Completely useless. The tumor needs a different tool entirely. It needs an anchor. And it gets that anchor through a mutation in a completely different network called the WNT pathway.
The WNT pathway, like the genes APAC or CTNNB1.
Exactly. APAC is the classic one.
So what does WNT actually do for the cell?
WNT acts like molecular glue. It creates what we call a block of differentiation. It literally glues the cell to the bottom of the crypt so it cannot be pushed up the escalator.
Oh, so it secures the beachhead. So the choreography here completely flips compared to the pancreas.
Right. Step 1 in the gut.
Yeah.
You absolutely must get a WNT mutation first to anchor yourself. Step 2, you can amplify KRAS later on to get aggressive and start invading other tissues.
And they saw this sequence in the lab.
They did. They screened these things called organoids, which are basically tiny mini guts growing in a petri dish. And when they looked at really early stage lesions called hyperplasia, they saw KRAS mutations, but no WNT mutations.
And what happened to those early lesions without the anchor?
They just vanished. They got pushed up the Velian shed. But when they looked at the next stage up, the adenomas, the actual established tumors, 100% of them had WNT activation.
Yeah.
You must have the anchor first.
Okay, but wait. What about the police? What about CDKN2A? If the tumor's growing and dividing at the bottom of the crypt, doesn't the police station notice?
In the intestine, the police are fast asleep.
Asleep?
Remember the chromatin we talked about? In the healthy intestinal cells, the chromatin around CDKN2A is marked with a different flag, H3K27E3.
Okay, H3K27E3. And that stands for?
Repressed. It means the DNA is packed away incredibly tight. It's locked in a vault.
So even if KRAS is screaming at top volume, the police literally can't hear it.
Correct. The alarm system is disconnected, so the tumor doesn't need to waste any time or energy trying to kill the police. And the data shows this loss of CDKN2A is really rare in intestinal cancer. It happens in maybe 11% of cases because it just wasn't standing in the way to begin with.
This is mind-blowing. It's the exact same gene, KRAS. But because the soil is different, a factory versus a conveyor belt, active chromatin versus repressed chromatin, the rules of the game, the actual order of mutations you need to survive, completely change.
Let's do a quick pulse check on where we are because that's a lot of biology.
Yes, time for recap number 2. Let's compare the notes. So pancreas. The barrier is the cell's identity. The police, CDKN2A, are wide awake. The rule, you must kill the police first, then amplify KRAS to force dedifferentiation.
Right. And the intestine. The barrier there is the conveyor belt. The police are asleep. The rule. You must anchor yourself with WNT first, and then you can amplify KRAS later.
Which brings us perfectly to Act 3, the lung, the middle ground.
Right, the alveolar cells in the lung.
What is the status of the police station in the lung, awake or asleep?
It's actually both. In the lung, CDKN2A is in what we call a bivalent state.
Bivalent, meaning 2 values.
Exactly. It has both chemical markers simultaneously. It has a little bit of the active flag and a little bit of the repressed flag. It's in a very low expression standby state, not fully alert and aggressive like the pancreas, but not comatose in a vault like the intestine.
So the lung cells are just a bit more tolerant of bad behavior.
Yes. They can handle high KRAS signals without immediately panicking and arresting the cell. So deleting CDKN2A is helpful for a lung tumor. It happens in about 15% of human cases, but it's not a strict day one requirement to start the fire. It's more of a late-stage booster.
Now, the researchers didn't just passively observe all this chromatin stuff. They actually proved this entire barrier concept with an experiment that honestly has the best name in the entire paper. Piggyback screen.
Oh, I love the piggyback experiment. It is so elegant. They used a transposon, which is also known as a jumping gene.
Explain how that works conceptually, because it sounds crazy.
It is a little crazy. Imagine a piece of genetic code that just randomly jumps around the mouse's genome, just hops from chromosome to chromosome, and every time it lands somewhere, it physically breaks whatever gene it lands inside.
Okay, so it's a random vandalism machine.
Yes. So they put this jumping gene into mice that were prone to cancer, and they just waited to see which tumors managed to grow out.
Ah, I see. It's basically forced survival of the fittest. If a specific gene is a major barrier, like a giant wall preventing cancer, the tumor absolutely must break that wall down to grow.
Exactly. That is the logic of a forward genetic screen. If you look at all the tumors that successfully survived and grew and you sequence them, you'll find where the jumping gene landed. You'll find the broken walls.
Okay, so let's look at the results. In the pancreatic tumors that grew, where did the piggyback transposon land the most? I'm guessing it landed right on the police station?
Nailed it. The #1 hit across the board was CDKN2A. It was a top 10 rank in 44% of all cases. The tumor absolutely needed the jumping gene to break that specific lock to exist.
And in the intestine tumors.
TDKN2A hits were super rare, only about 9%, because breaking in a sleep police station doesn't help you. Instead, the top hits where the transpose landed were all WNT pathway genes.
That is such incredibly clean proof. I was thinking about an analogy for this. It's kind of like checking the toolkits of a bunch of successful burglars in different neighborhoods.
Oh, I like that.
Right. Like if you check the burglars in neighborhood A, and every single one of them is carrying a heavy-duty glass cutter, you know for a fact that the houses there have a window barrier. But if you check neighborhood B, and they are all carrying lockpicks, you know those houses had locked solid doors.
That is a perfect analogy. The pancreas has a CDKN2A window that must be shattered. The intestine has a WNT door that must be picked. The tools dictate the barrier.
So what does this all mean when we zoom out? We've looked at the MCCA tools, we've looked at the KRAS volume knob, and we've toured the organs.
It brings us to the ultimate synthesis of the paper, the big so what. This paper powerfully argues that cancer evolution is not just random chaos, it is deterministic.
Deterministic, that is a very strong word to use in biology.
It implies true predictability. It means that cancer isn't just a drunk guy stumbling through the dark. It means if you give me 3 specific variables about a healthy tissue, I can predict the exact genetic trajectory a tumor will have to take to grow there.
What are the 3 variables in the formula?
Variable 1, the cell of origin. Are we dealing with a static factory worker like the pancreas or a fast-moving conveyor belt like the intestine?
Okay, the mechanics.
Right. Variable 2, the epigenetic state. Is the chromatin at the major tumor suppressor loci open and active, or is it closed and repressed? Is the police station awake or asleep?
And variable 3.
Tissue dynamics. Does the tissue naturally shed cells, or does it hold on to them tightly?
So if you plug those 3 things into the equation, it spits out the exact order of mutations the cancer has to acquire.
Correct. It defines the choreographic steps. And the amazing thing is, we only know this now because they built the MCCA. We needed those 590 specific clean maps to compare these tissues side by side without the noise of the brown mouse ghosts.
It really changes how you fundamentally think about the big C. It's not just awful bad luck. It's a highly logical, almost mathematical sequence of breaking very specific barriers that exist in our healthy tissue.
And that realization leads to the final and most exciting takeaway.
Okay, let's summarize the big beats first before we close out with that thought. Just a rapid-fire rundown for you guys.
Yeah.
Beat 1. The MCCA resource finally fixes the immunophenotyping blind spot. If you're a researcher, watch out for those 129 strain passenger SNPs in your black 6 mice, or your drug trial is basically toast.
Beat 2. KRAS dosage matters. It's not a switch. Tumors fight tooth and nail to reach IgD, increased gene dosage.
Beat 3. In the pancreas, the barrier is CDKN2A. You must delete it early to allow high KRAS to force the cell to dedifferentiate.
And beat 4. In the intestine, the barrier is the high cellular turnover. You need WNT to anchor the cell first. CDKN2A is completely irrelevant because it's already repressed.
It brings us right back to the intro. The mutation obviously matters, but the tissue context is what writes the rules of the game.
Exactly.
So I want to leave you with one final kind of provocative thought to chew on. Building on what we just learned, if we know that the soil, the specific epigenetic state of our healthy cells, is what allows or prevents cancer from taking hold, could we therapeutically lock the chromatin of our healthy tissues?
That is a billion-dollar question right there.
I mean, think about it. If we could use a drug to ensure that CDKN2A stays wide awake, permanently stuck in that H3K4ME3 active state in the lung, or if we could ensure that differentiation is absolutely unstoppable in the intestine no matter what WNT does, could we make our tissues evolutionary dead ends for cancer initiation?
It's a profound idea. Instead of just waiting for the mutation and trying to drug the bad protein, we proactively reinforce the structural barriers of the house. We make the soil entirely inhospitable to the seed. It's a totally new preventative strategy that this paper really opens the door to exploring.
It really does. And for the scientists and researchers listening out there, I want you to think about that effective PTMB concept we discussed, the ghost in the machine. How many past drug trials, maybe even your own, might have failed or falsely succeeded because of hidden genetic mismatches in the mouse models?
It honestly makes you want to go back to the lab and sequence every single cell line currently sitting in the freezer.
It really does. It forces us to question our most basic assumptions. But hey, that's what we do here. Thank you so much for joining us on this deep dive into the choreography of cancer.
And as always, keep questioning the data.
We'll see you next time.